Hello, and welcome to the first in the lecture series on DNA, genetics, mechanisms of microbial genetics, all the above. We're going to get into the nitty-gritty of the central dogma and DNA, RNA, and protein, and what it all means for bacteria. Right? So pay attention to these learning objectives as we go through the video. We'll go through a case study right now, and then we'll go through some of the subjects on DNA and microbial genetics. First, we'll go into DNA, and then RNA and how we'll compare them. We'll see the main functions of them, and then we'll get into how it's actually replicated, and the proteins and enzymes that we need to do that. We had already learned about enzymes before. And then we're going to learn about mutations at the end, and what types of mutations there are. And we can compare them to like the X-Men mutations, maybe, just to lighten up things. And then we can also discover how microbes, due to the fact that they cells, so how do they maintain their genetic diversity? And so we'll learn about bacterial conjugation and how to use their structures to do that. Right, so moving into our case study, after we learn about that the same DNA make up a elephant and a bacteria. Okay? We use the same DNA to RNA to protein mechanisms that are present in microbial genetics. We use them in elephant genetics, human genetics, animal genetics, and everything in between. It's all the same DNA, RNA, protein, and that's why we learned it way back when. We learned about biomolecules, organic molecules, and we need all of those parts in order to do this. We even need lipids in order to contain all of our genetic blueprints. Right, so hopefully you've done like all the other case studies and have actually read ahead on these case studies, so I don't have to go into all of this, but basically Mark has a pre-existing condition of type 2 diabetes, and he does some gardening, gets himself cut on his leg, and becomes red, increasingly red, even warm to the touch. Right? So after 3 days, finally, he develops a fever, stiffness in his leg. He goes to the emergency room. Okay. Did Mark wait too long to seek medical attention? And at what point did his signs and symptoms warrant medical attention? Okay. It's hard to say, but if you have pre-existing conditions like type 2 diabetes, you might want to go and see the doctor. If your wounds become red, you might want to take care of it first yourself with soap and water before you just jump to conclusions. But once it becomes warm to the touch, swollen, and increasingly red, right, instead of decreasingly red, it was time to go to the doctor. Okay, so waiting this extra 24 hours did Mark no favors. Okay. And what we just pointed out, red, swollen, warm to the touch, those would all be signs, and those are measurable. And symptoms would be, I feel poorly, stiffness in the leg. Those would be symptoms. Because everything else we can measure. We can measure the redness, measure how much it's swollen up, compare it to the other leg even, and even feel the temperature of the leg. Okay. We can also measure the temperature of fever. Okay. So the stiffness in the leg would be a symptom. What other types of infections and conditions might be responsible? We went over that. This is type 2 diabetes could give you some stiffness in your leg, but probably not all those above. Especially since it's at the wound. You want to think that it's not as diabetes, that it is actually probably a bacterial infection that he received from plant or stick or piece of dirt that he got into his wound. Okay, so he gets to the emergency department. Nurse tells him this got out of hand, he waited too long. Skin progressed, everything progressed to necrotizing fasciitis. And basically that means it's going to eat your skin. And they ordered a culture fluid that was draining from the leg, ordered blood work, white cell count. Okay. And even though he was treated with some antibiotics, he deteriorated quickly, became dizzy and bloody. Okay. So within a few hours of his hospital admission, his blood pressure drops, breathing slow, develops all of these other signs and symptoms. And what does that mean? It means he waited 24 hours too long, right? This is hour 28. He could have been here at hour 8. Okay. So what are the possible causative agents of necrotizing fasciitis? We're probably going to go bacterial. We're going to think bacterial. But hey, wait, prescribed antibiotics. What's happening? Well, they are resistant. So let's test for MRSA like we did in the lab. Let's test for any other kind of resistance. We can do those tests. What are some possible explanations for why the antibiotic treatment does not seem to be working? Well, what are we learning about in this chapter? Right? Learning about genetics, learning about DNA, RNA to protein. Well, we probably have some kind of DNA sequence that makes some RNA, makes a protein, that makes it so that we either can digest the antibiotic resistance, or our protein is shaped in such a way that it's not going to be infected by the antibiotic. Okay, so it's resistant. Okay, so we're going to make a resistance protein or a resistance toxin, and we're going to see all about it. All right, despite continued antibiotic treatment, continued to progress, and ordered surgery. Okay, they're going to debride all of his wound. Okay, after 2 more surgeries, they finally seemed to be contained, but they needed to gouge out his leg, right? They took out a lot of skin away, they took a lot of muscle away, and so now he's got a partial leg. Okay, but he's alive, and it wasn't amputated. Okay, so there's that. What is the removal of the infected tissue from infection? Okay. It's like The Walking Dead, right? Like when you cut off your arm right after you get bit, you can get infected. Same thing. If you can contain a bacterial infection because it didn't get into your bloodstream all the way, and it's just contained at the wound. How do you know it's contained at the wound? Well, it hasn't spread, right? It's just swelling around the wound. It's just puffing at the wound. So because it's just there, we can just start removing all the tissue. But we gotta go deep because see it and get that last little part. Are there like complications with this method of treatment? Well, muscle and skin mass. Okay. And eventually you could lose the leg if it goes to the bone. Okay, lots of words. So again, pause the video, read it ahead of time. Make sure you understand what I'm talking about. Okay, so he survived. Okay, lost a lot of muscle mass like we talked about. Physical therapist is optimistic. Laboratory testing did confirm resistant streptococcus. Okay, and as necessary, it needs to get reported to the CDC. Now, we did that methicillin resistance test, the MRSA test in the lab. You would not be sent to the CDC. It's just for informative reasons. What this is saying is when you're infected and sick, and that is caused by MRSA, we have to have those three things in place, then it would get reported. Okay? So methicillin resistance is genetically encoded. This is why we're learning about it. We're going to learn about that later in the chapter, all the way down, where literally you can imagine them just horizontally transferring the genes between two cells. We're going to learn about it when we talked about the epiphylus and the cell structures. Okay. Anyway, they genetically encode bacterial toxins. Those toxins limit the effectiveness of antibiotics, and so his treatment wasn't working. So he was continuing to harbor the bacteria and was continuing to grow and divide it. Okay, and so Mark's case had several genes on his propages. And the propages, as we learned, come from the viruses, right? Okay. So propages are the latent infection of viruses in bacteria. Okay, and it's viral DNA inside the bacteria. And it can be on plasmids, but since it doesn't indicate as such, I wouldn't say that it is. It's probably in its actual genome. Okay, and so with that, we'll get into the structure and function of DNA and RNA in the next video.